Welcome to Cube Biotech's scientific masterclass series, where researchers from around the world present their latest results and findings to a global audience. So my name is Michael Erklens, and I'm part of the research and development team of Cube Biotech. So today I'm happy to introduce this masterclass series, which provide a unique uniform platform for scientists to share their work, connect with others in their field, and receive feedback from experts and peers around the world. After the talk today, there will be time for the interactive question and answer session. So during the talk, you will be able to use a chat function to comment directly aspects of the talk, or you can place a question for the Q&A session by changing the chat mode to the Q&A mode. So you can see it on the bottom right of your screen. Our speaker today is Professor Bert Klumperman, who is currently professor at the Stellenbosch University in South Africa. So he's also founder of the spin-off company Nanosene. So now I would like to introduce his CV. And now you can also see Professor Klumperman. So hi, nice to meet you today. So Professor Klumperman was born in 1962 in Hellendoorn, the Netherlands. So he studied chemical engineering at the University of Twente in Enschede. And in 1994, he obtained his PhD from the Eindhoven University of Technology, so under the supervision of Professor German. In the next year, Bert Klumperman joined the Eindhoven University of Technology as an assistant professor, where he was promoted to associate professor in 2002. In 1998, he came in contact with the Institute for Polymers Science at the Stellenbosch University. In 2008, he was elected Fellow of the Royal Society of South Africa. So in the following years, he received several prizes, and I would like to mention a few of them. So in 2015, for example, he was awarded with the SASOL Chemistry Innovator of the Year medal. And in 2016, he received the National Science and Technology Forum Lifetime Award. Also in 2018, for example, he received the John FW Herschel Medal of the Royal Society of South Africa. And from 2021, he is now also Vice President of the Royal Society of South Africa. Additionally, in 2012, Professor Klumperman was awarded with the second doctorate at the Stellenbosch University with Professor Mellon as a promoter. So, and maybe also for us at Cube Biotech, the most important thing, since 2022, he is a Chief Technology Officer of a spin-off company called Nanosene, which should commercialize the polymers he developed in his research group. And we are also we launched also some of their great products. But I think he will refer to them later on. So his current research interests are reversible deactivation radical polymerization processes and the SMALP technology in general, and the biomedical applications of the smart copolymers for the use around the world. So I hope I did not forget anything, and it was comprehensive. So with this, I would like to hand over to Professor Klumperman and his exciting talk today. So thanks for your attention from my side.
Thank you, Michael, for the kind introduction. Let me quickly get the slides on. Here we go.
Great, thank you.
Yep. So I would like today to give an overview of the SMALP technology for the isolation of membrane proteins, and specifically on the role of the amphiphilic copolymers in that process. I will try to do this by discussing some of the characteristics of the first generation SMA copolymers, and then by explaining how more advanced polymerization techniques provide access to various features that are unattainable by the traditional process. Towards the end of my presentation, I will then give some examples of how I think the potential of second generation SMA copolymers can be unleashed. I will start off my presentation with an animation, and that was not possible in the presentation, so I will quickly open that. Let me see. Here we go. So This shows how we believe the SMALP process takes place. Polymers start to interact with a membrane fragment, and then penetrate the hydrophobic parts, penetrate into the hydrophobic layer of the phospholipids, and then you create these nanodiscs, and inside the nanodiscs you will have your proteins that can then further be purified. So what about these the polymers that are involved in this process? Let me quickly, this is the animation. So this is the end product that we're looking for. We've got the membrane protein with the annular lipids, and then surrounded by the amphiphilic copolymer, where usually it is assumed that the hydrophobic parts of the polymer are penetrating inside the hydrophobic layer of the phospholipids, and the hydrophilic layer sort of stabilize the whole construct towards the aqueous phase. Of course, as you probably all know, the original work in 2009 by Overdijn and Deforn reported on this polymer, SMA 2000 at the time, from let me see, that was still from the US company that is no longer providing the SMA copolymer. It has got a styrene to maleic acid ratio of 2:1 and number average molecular weight of about 3,000 grams per mole. The dispersity is quite large. It's about 2. And I will refer to this material as the first generation SMA copolymers. It's a family of polymers. Also, the 3:1 is used, but I would say the working horse traditionally is the 2:1 version. So I want to zoom in a little bit on the molecular weight distribution. What does it actually mean? So here we've got a molecular weight distribution, as most of you will probably recognize it, on a logarithmic x-axis, so ranging from 100 to 100,000 molecular weight. And then I've drawn here curves of polymers with different dispersities, going from the very narrow one, 1.08. Let me see if I can quickly switch on my pointer. So the very narrow one, the light blue one, and then all the way to the 1.84, which is the black curve, and everything in between. So the traditional polymers that you make via radical polymerization, conventional radical polymerization, will have a broad molecular weight distribution, a dispersity in the order of 2. And what does that look like? This is the chain length distribution of the Cray Valley SMA 2000. Like I said, average molecular weight of 3,000, and log molecular weight on the x-axis again, and the weight fraction on the y-axis. So this is what everybody knows. Now, if you look at this distribution in a slightly different way, namely the so-called number distribution, you will see that actually a large fraction of the polymer chains have a very low molecular weight. Now, there's a linear x-axis, but you can see that the majority is actually of very low molecular weight. Why does that happen? Well, this is completely due to the process by which you form the polymer chains. You continuously create new radicals that have a very short growing time of about 1 second or so. And during that growing time, each time they have the option to either propagate or terminate. And the ratio between the two is completely based on probability. So you get a stochastic distribution of chain lengths, and that eventually leads to a distribution as you see it here, and as you saw in the previous slide in the normal log molecular weight distribution, with a dispersity, as I said, of 2. Now, if you want to have better control over your polymer chain length and other features of your polymer, how do you do that? Well, I'll try to explain that by this following scheme. In this scheme, you will see the steps that are done in a conventional radical polymerization initially. So we've taken an initiator, and it decomposes into 2 radicals. These radicals add 1 monomer to form the start of a growing polymer chain. Then repetitively, you will do this propagation reaction where a monomer adds to the growing radical, and you form a chain of a specific length that then, as soon as it encounters another radical, will terminate to form a chain, either one long dead chain or two original dead chains, depending on the termination mechanism. I won't go into the details of that. So to give you an impression of the time scales of these reactions, I've drawn down two mathematical equations here at the bottom. The first one shows you what the characteristic time is for a propagation step. What does that mean? Well, the propagation rate constant times the monomer concentration gives you a frequency. There's the frequency of monomer addition to a polymer chain. And the reciprocal of that is the average time it takes for one monomer to add to a polymer chain. So I've filled in typical numbers, say 2000 for your propagation rate constant, 5 moles per liter for your monomer concentration. You end up with 10,000 here in the denominator. So you get a characteristic time of 10 to the minus 4 seconds. So every 10 to the minus 4 seconds, a monomer adds to a polymer chain. So then how long does it take for the polymer chain to terminate? Well, you do exactly the same thing, but now for the termination reaction. You take the termination rate constant times the radical concentration. This will be in the order of 10 to the 8, 10 to the minus 8 for the concentration. So you end up at 1 second. So that means that a polymer chain, after it's initiated, will add a monomer on average every 10 to the minus 4 seconds, and after 1 second on average, it terminates. So you have created an average chain length of 10,000 monomer units in this case. So this is a polymer chain that's significantly longer than we use for our current application. But this is problematic because on a time scale of 1 second, there's very little you can control. So in the 1990s, researchers around the world started to sort of get an idea of how to bring this process under control. And that was done in a general sense by adding this, a deactivation step. So you've got your radical that is growing at any stage of its chain growth, and it can add to a compound X to sort of put it asleep, make it into a dormant chain, and it can be reactivated. So the reactivation step is not so important. Of course, it needs to happen because otherwise your polymer is dead, but it just determines the overall rate of polymerization. But the deactivation step is very important because you need to be on the time scale of the propagation step or faster to exert control over your chain growth. So we can do exactly the same as we did here for propagation and termination, we can now for deactivation. So 1 over deactivation times the deactivator concentration. This must be a very fast reaction. So the rate constant is 10 to the 9. That's a diffusion limited reaction. The concentration, you can choose 10 to the minus 4 mole per liter. And you end up with a characteristic time of 10 to the minus 5 seconds. So it's even faster than your propagation reaction. So it means that your chain will deactivate, reactivate, deactivate, and then occasionally in that continuous activation and deactivation, add a monomer every now and again. And in doing so, you're able to let all the chains grow simultaneously, and you end up with a narrow molecular weight distribution. So the technique that we use for the largest part, we also use other techniques, but this is our working horse, so to speak, is a raft-mediated polymerization. And this is a typical raft agent, as we call them. It's a reversible chain transfer agent, and it's got two important components. The one is the so-called R group, which is the leaving group radical, which sort of should start the growth of a polymer chain. And the other side is the Z group, and the Z group determines the reactivity of the raft agent and the stability of an intermediate form of the radical adduct, which I will not be discussing today. If you do all of that correctly, what you will be doing is insert monomer right in that carbon-sulfur bond. And eventually you will end up with a chain that is shown here. In green is that former R group. Now, if you've got your polymer chain, in this case, a styrene maleic anhydride copolymer, so that's before hydrolysis towards SMA, and then at the other end, you will have your raft functionality. So what have we achieved if we do the polymerization like this? Well, first of all chains grow simultaneously. That leads then to a narrow molecular weight distribution, and we can get up to 95% chain end fidelity or even higher. And we have the possibility to create block copolymers. Once all the monomer has run out, we can add new monomer and insert monomer here in this carbon sulfur bond. And if there is a different monomer, then you create a block copolymer. So there are many more variations possible by using this controlled radical polymerization technique. So what we have done is we have made a mimic for non-alternating SMA. I will not go into the details of that today, but it is complicated to make via raft polymerization a non-alternating copolymer. Making the alternating version is actually quite straightforward. So it's easy to make a narrow molar mass distribution polystyrene alternating maleic anhydride. But the problem is, as many of you know, alternating SMA is actually too hydrophilic. It does do a bit of a job in isolating membrane proteins, but not nearly as good as the 2:1. So what we then decided to do is partially modify the polymer to convert some of the maleic anhydride into a hydrophobic unit instead of hydrolyzing it to maleic acid, which will make it hydrophilic. And we end then up with this polymer here, which is maybe slightly complicated to recognize, but here's one styrene unit, and here is one modified maleic anhydride, which is now a benzyl polyamide. Here's another styrene unit, and the maleic acid with the two carboxylic acid groups. And there is a fraction F of the maleic anhydride that has been converted into the polyamide, and the rest, 1 minus F, is your hydrolyzed maleic acid. So we can get a controlled and narrow molar mass distribution polymer. We can use F to control the amphiphilicity. And here in gold, I put something which still holds future for the promise for the future. We can introduce chain and functionality, which is something we have not explored in great detail in the current products, but we are working on that as we speak in my research group. This product is now commercially available via Q Biotech under the trade name SMALP BZ. That is this polymer, and that was developed by Giselle Keuler in my group, and a patent is pending on that material. So just to show you some results, this polymer has been tested on a number of different proteins. I will show you two. First of all, a GPCR, the human neurotensin receptor. It's involved in the regulation of blood pressure, and we have expressed it in E. coli in the group of Mark Wheatley. It got a monomeric unit of 46 kilodalton, and it usually is present in its dimeric form with a 92 kilodalton molar mass. And if you then look at the SDS-PAGE results, you can see that if that factor F that I showed you in the previous slide, if you increase it from 0.05 all the way up to 0.40, you can see here in the soluble fraction that initially the polymer is too hydrophilic to actually isolate the membrane protein. And here from 0.20 onwards, you start to see that there is some protein being present, and it increases all the way up to 0.40. If we modify the polymer beyond 0.40, the polymer is no longer water soluble, which means that it's no longer suitable for this application. You can also see from the intensity of the band for 0.40 that it is certainly as good as DIPMA or SMA 221, but you could even argue that it's slightly darker than those two. Similar story, of course, for the insoluble fraction. For the very hydrophilic polymers, you see that there's a lot remaining in the insoluble fraction, and less so for the more hydrophobic ones, and for the DIPMA and SMA 2:1. Remarkably, what we see in another polymer, KCSA, the ion channel, which is a tetrameric protein. I've shown some pictures here, and if we look at the SDS page of this system, you can see that it's actually different, very different. You can see here is the insoluble and soluble each time next to each other for the SMA 2:1, 1:1, and then starting from the different F values, 0.05, again up to 0.40. Now you can see that 0.40 actually has the faintest band, and that there is arguably a sort of a maximum maybe around 0.15 or so. But clearly there is a very different dependence on the degree of modification for this protein compared to the previous one. And we believe that to be caused by either the nature of the protein or probably even more likely the annular lipids, so the closely associated lipids around the protein. So in summary, and then I will continue with some of the future outlook and that potential of the second generation polymers. The first generation SMA copolymers have a broad molecular weight distribution, and it's got limited possibility for functionalization. You can only do random functionalization, as many of you probably know. SMASH, the polymer that was published several years ago from the group of Marie Aubin-Tam at the University of Delft. But for the second generation SMA copolymers, we've got a narrow molecular weight distribution, and we can do both in-chain as well as chain-end functionalization. And that allows us, we think, to make tailor-made polymers for very specific applications. And I want to show you two examples of that. Oh, no, first I want to show you what it actually entails. I was speaking about that R group. That R group, in my previous explanation, was just sort of an arbitrary group that is suitable for reinitiating the polymer and for forming that chain end. But you can easily envisage that you can include something there, be it an affinity tag or a fluorescent tag or something like that. Then what you can do, then on the other hand, of course, you've got your RAF moiety still there. That RAF moiety is easily modified post-polymerization and allows you to introduce a second functionality. Again, can be affinity, can be a fluorescent tag. You can think about thread pairs between the one side and the other side. There are many, many possibilities that you can think of. And of course, we can also do on our polymers what can be done on the commercially available traditional polymers, and that is random modification along the backbone of the polymer. Then to show you two examples that we're currently working on. This is the work of Lauren Ball in my group. She's working on block copolymers where the SMA mimic has now a hydrophilic block connected to it that allows you to immobilize a nanodisc sort of close to a surface. We're thinking obviously in the direction of gold surface for SPR applications with this. And another one that we're currently working on is, you can clearly see, I hope that this is very fluorescent. So what we have here is a polymer that also contains a little block as a spacer, and then a fluorescent tag at the chain end. That spacer doesn't necessarily need to be there, but it just shows you how you can play with the different structures that you can create for the various applications. It rests me to thank some people, because all of the work that I've been talking about and the explanations only come about by having a fantastic group of students working in my lab. They're listed here, some of my postdoc and a collaborator, and the rest are students in my group. Then we are working on a Wellcome Trust grant currently, and that is in collaboration with a group of Prof. Tim Deffern at the University of Birmingham, and various members of his group are also collaborating with us in this project. And then the work that Giselle has done on the GPCR, or done in the group of Professor Mark Wheatley at Coventry University. And also there we work with other members of the group. Rebecca is actually a joint student between the Coventry lab and my lab. And the funding initially always came from the National Research Foundation in South Africa. It's currently coming from the Wellcome Trust. And I want to thank Q Biotech for giving me this opportunity, and also for being a partner in the commercialization of our polymers. And as Michael already indicated, we have a company that is also looking at commercializing polymers that come out of our research group. That's Nanosene. If you're interested, here are the contact details. And with that, I'll be happy to answer some questions.
Great. Thanks a lot for this exciting talk. So it's really, really impressive. And I think this, also the functionalization and the tagging or the adding of a fluorophore to the polymer will be really a great step forward, right? So especially for downstream applications, it's it's simply necessary, right? So just to track your nanodisk, or just capture your nanodisk with a tag. So it's a great thing, and we are looking forward to your new techniques. So with this, we can start the interactive question and answer session. But I will ask the first question.
Sure, go ahead.
It's good to be the presenter, so I do not have to wait. Yeah, right. So something which I hear quite often from the people is why is the low polydispersity of the polymer so important? I mean, compared to, for example, SMA 200, commercially available, it also works. But what exactly are the benefits of the low polydispersity for the user? So, for example, simply a biologist would like to solubilize a protein and purify it.
Sure.
What do you think?
Sure. So if you it depends a little bit on how you use it. If you take the isolate, if you make the nanodiscs of a membrane prep, say, and you then run immediately a gel, then you will very often see that with the broader molecular weight distribution that you get smearing on the gel. So that is something you can quite nicely avoid if you use the narrow molecular weight distributions. On top of that, I think that the low end of the molecular weight distribution of the commercially or the traditional polymers may have characteristics not too dissimilar from low molecular weight detergents, and with that you can also get the associated negative effects. You know, that you start solubilizing lipids rather than making nanodiscs. The high end of the molecular weight distribution, on the other hand, can do other unwanted things. You know, you can get bridging of nanodiscs by, you know, one part of the polymer sitting on one disc, one on another disc, and increasing viscosity and causing problems in that way. So those are the general negatives, or you could also say advantages of narrow molecular weight distributions. In a more general sense, I think if you do scientific research, especially, then having a well-defined material is always better, because then you can, you know, really look at the influence of one specific parameter, and you don't need to worry about, you know, but I've got all these molecular weights in my mixture.
Yeah, it's really interesting. So maybe a connecting question. So people sometimes wonder that more than one polymer chain is involved in forming the nanodisc. So the bridging you mentioned before. So, and we're quite surprised. So, oh, wait, so there are more than one polymer in the nanodisc. So what you tell us, so if you use a well-defined polymer with a distinct size, so you choose, for example, I think you screened the size, or the right size, the best size for your polymers. So then you can avoid this, right? So
well, I think that you won't avoid that you have more than one polymer chain per disc, because the chains are simply not long enough to you probably need a couple of rotations around the disc to properly stabilize it. and the chain is not long enough. If you would make it long enough, then you get bridging between two, and then you get other problems. So I think it's unavoidable that you use multiple chains, but until now, we don't know exactly what the number of chains is. One of the things on our wish list to sort of do, once we have to fluorescently tag polymers, you can obviously start counting the number of chains on a disk.
Oh, great. So this will also be a great step forward. So let us know. Exactly. So it's a really interesting question, and also quite valuable, right? So let's start with the question and answer session. Right. So the first question was asked by Bert Klumperman, and he asked, is it possible to use copolymers and lipids to stabilize cell-free expressed membrane proteins?
That's a very good question. So we're polymer chemists, so we don't really do things like that. But my impression would be that you could. I mean, but it then goes more in the direction of what people do with the scaffold proteins, right? The MSPs, because it's almost like reconstituting the membrane proteins in an artificially made disc. I don't think that the copolymer, I mean, they will probably work for that purpose, but I don't think they will have a great benefit over MSPs, apart maybe from the cost.
So maybe I can also give an additionally answer. So, for example, there were some publications recently. So, for example, the sulfo-SMA and sulfo-dipmer polymers, they're used for the cell-free expression of proteins, and the benefit of the polymers is, for example, the stability to magnesium or calcium. So this can be, for example, can be quite beneficial with respect to copolymers. So you can use them, and there are polymers available for this. Yeah, so the next question was asked by Hugo Henderson. So did you have a specific application in mind when designing the SMALP BZ copolymers? I mean, follow-up applications after the stabilization?
So the main reason for us to make this series of polymers is that we wanted to have a series of polymers that just vary in one parameter and one parameter only. So all the chains have exactly the same molecular weight, the same everything, except that they vary very systematically in hydrophilic hydrophobic balance. And the idea behind that is that we then wanted to look into that specificity that I was alluding to a little bit when I showed the two examples, that the one has a different optimum degree of modification compared to the other. So that was the actual. So it's more like almost like a research tool from that point of view, that you can start to probe the selectivity of the polymers for isolating a specific class of membrane proteins.
Okay, great, thank you. So the next question is asked by Melissa Eaton, and she's asking, how did you stabilize NTR run from the bacterial membrane system? I thought bacterial membranes were a no-go for copolymer solubilization.
So that part of the work we did all on membrane expressed proteins. And again, I'm not a biologist, but as far as I understand, the polymers work. Well, we've shown that they work on bacterial membranes as well as on mammalian membranes. So I don't know what else to say about that. It's actually the system works as far as we can tell universally between bacterial and mammalian membranes. You potentially just need different degrees of modification for the reasons that I just explained.
Yeah, I think it also depends strongly on the polymer use. So if you try, for example, the whole set of polymers, so like the solubilization kit, so then you will also maybe find your polymer working for your bacterial culture or your protein of interest. So, but actually, I'm also not quite in this in this part. So, yeah, maybe. Great. So the next question is asked by Cecilia Canessa, and she's asking why affinity purification is low yield with SMARs and high protein losses on SEC.
I think that varies a lot on the exact system. We have seen some systems. I think, Michael, you also indicated once that it's very variable, right? From one system to another, the amount of loss that you get in your purification. We have seen some systems where we don't get we get quite good yields, but other systems where the yield is not so good. So I think that is very much system dependent, and whether that is directly related to SMA or to the protein, I'm not 100% sure.
Yeah, I totally agree. So as a colleague from Cube said that a few months ago, so screening is key. So also you should try then different polymers or different BZ SMARs, for example, or simply also different SMARs itself. So then you can also avoid high protein losses on SEC. So it can be a simple answer like, so avoid divalent cations or use another ion strength of your buffer system. So there are multiple solutions, and you have to screen it, I think. So there's no simple answer for this, I think. Yeah, great. So the next question was asked by Ruth Karis, and she's asking regarding modifications, could you add an affinity tag to the polymer and subsequently purify the membrane proteins this way without tagging them themselves?
Yeah, so that's a very good question. Yes and no. And so you cannot get a specific protein if you tag the polymer, because obviously the polymers will not, the protein and polymer combination can be different. I mean, if you take a membrane, you solubilize the entire membrane, all the nanodiscs will have the tagged polymers. So that means that you will be able to purify nanodiscs from other material, but not specific proteins. So I think the affinity tag is more for the immobilization than it is for actual purification of the membrane proteins.
Yeah, so I think you will also isolate all the other membrane proteins and empty discs, right? So maybe also, or maybe only for the pre-purification from the cell debris. So
yeah, exactly.
Then maybe, but yeah, I think you will also isolate the other stuff from the membrane.
Yeah,
can be difficult, but of course, so some proteins do not have a tag, or it's not possible to tag them. So maybe it's the best you can do to simply avoid debris.
No, certainly. If you're working with wild type membranes, so that there is no modification of the membrane proteins, inclusion of his tags or what have you, then it's the best you can do, and you will have to look at alternative methods for the purification. But there will be no selectivity in that sense, because, like you said, all the discs will have the tag.
Okay, great. So the next question was asked by Bernd Löhnemann, and he is asking, do you see any advantages of free radical polymers over raft polymers, maybe for very large protein complexes for which the broader molecular rate is more handy?
Well, I already mentioned my view on the advantage of having a narrow molecular weight distribution. We have not yet really investigated of whether it's beneficial to have higher molecular weight polymers for larger complexes. My gut feeling is that the answer will be no, because you don't have, like I said, you don't have single polymers per disk. and you will always have multiple polymers that sort of sit around the disk. I think potentially the only advantage of free radical polymerization is that it's easier to do, cheaper to do. I've said for very different applications, I always say, if you can make a material without having to use controlled radical polymerization, then you should definitely do it. I mean, it will never, raft will never replace, raft made polystyrene will never replace the polystyrene that we use for coffee cups, because it's a commodity product, and by raft you almost inevitably make these sort of specialty products, high-end products.
Okay, great. So there are questions left, but I think we are running out of time. So, but don't worry. So the questions which will be, or which are not answered today, will be answered by mail. So don't be worried. But I will ask the last question. So, how important, what do you think, how important is the flexibility of the benzylamine moiety during the solubilization or in the polymer itself? So I was wondering, so you specifically choose benzylamine as a modification for the polymer. But what do you think if you use, for example, benzylamine with a kind of linker between the polymer and the styrene ring? So what do you think? Is flexibility in the hydrophobic part of the polymer beneficial for the stabilization or the penetration in the membrane during the solubilization? Or what do you think about this topic? Because I saw lots of polymers in the last years, and they differ quite a lot, right?
Yeah, so I think that so the reason why we chose benzylamine is that we wanted to mimic the phenyl ring of the styrene and make it sort of almost as close a mimic as possible. But it's definitely not the only one you can use, and I'm convinced that if you use other amines, hexalamine, but also aniline, that it would not make, it would be different. It would be different, and there would be a different optimum in terms of the level of modification that you need. Specifically about a spacer in between the phenyl ring and the attachment to the backbone. I think that probably depends a little bit on whether it will be a hydrophobic or a hydrophilic spacer. If it's a hydrophilic spacer, you get something that's very different. It would be interesting to look at that. If you get a hydrophobic one, the whole construct may become very quickly too hydrophobic, and start self-assembling. So the system, the polymer, of course, is relatively low molecular weight, in the order of 5,000 or so molecular weight. If you include large hydrophobic groups, we've seen also from other experiments that it's quite easy to get aggregate formation, micelle formation. Even if you use raft agent that have this long C12 tail, like some people do, you will immediately see that you get micelle formation of the polymers. So it's difficult to predict, but I'm convinced that benzylamine is definitely not the only option, and we are actually exploring other avenues from that point of view as well.
Yeah, I mean, especially for the drug targeting or something like this, so then it can be maybe quite good to form particles, but for the solubilization, maybe not.
Sure.
Not so
good.
Yes,
100%, yes.
Yeah, great. So, thanks a lot for this exciting talk and the great question and answer session. So it was really a pleasure to have you here, and it's a cutting edge topic, right? So and we are looking forward for your publication in the next month and year. So
yes,
I'm sure you're working on more also in your company, so in Nanosene. So we are really looking forward.
Well, thanks for having me. It was very nice to give this presentation and answer the questions.